Now let's talk about another important thing, that is ribosome. So ribosome is a macromolecular machinery very important for protein synthesis. So any cell who requires protein should have ribosome for the synthesis of protein. So this ribosome is consist of two subunit, large subunit and small subunit, that together assembled to form ribosome. Remember three things: ribosome, mRNA and tRNA. These three things together call as transcriptional apparatus because they together work to produce protein. How? Let me give you a overview. So what happens if I try to explain it from the DNA level? So let's consider this is DNA. So from DNA, mRNA will be synthesized. That is the messenger RNA. And why we are calling this type of RNA as messenger RNA? Because here every successive 3 nucleotides will be considered as one codon. And this codon will determine which amino acid will come and when. Okay, for example, let's say in most of the cases at the first 3 nucleotide will present, that is A, U, G, and this 3 codon signifies methionine amino acid. That's the idea. So what will happen that in the, and remember the entire thing will be going on in the ribosome. So this mRNA will come and then tRNA, which is another RNA, and the tRNA is transfer RNA. So what tRNA will do, remember mRNA have codon, similarly tRNA have anticodon. Okay, so tRNA will going to read the codons present in the mRNA, and depending upon what is present, it will going to bring amino acid from the cytoplasm. Okay, tRNA have a amino acid binding site with the help of which it will going to bring the amino acid, and then it will bring depending upon what type of codon present at that particular mRNA. Okay, and that's how depending upon the codons present in the mRNA, amino acid will come. And then in between them formation of peptide bond occurs, and this peptide bond formation is especially done by ribosome. Okay, so now you may understand. You can see here the peptide bond is forming, right? So that is the idea, that is the main role of ribosome. Okay, so I have told you that ribosome consists of large subunit and small subunit, they together assemble to form ribosome. So in case of prokaryote and eukaryote, in both the cases ribosome are present, and in case of prokaryotic ribosome and eukaryotic ribosome, these two are significantly different. Okay, that means prokaryotic ribosome have a remarkable difference with eukaryotic ribosome. So let's discuss what type of difference are present in prokaryotic ribosome and eukaryotic ribosome. So in case of prokaryotic ribosome, the sedimentation coefficient is 70s, and in eukaryote the sedimentation coefficient is 80s. Okay, now in prokaryote it have large subunit and small subunit. Similarly in eukaryote it have large subunit and small subunit. In prokaryote the sedimentation coefficient for large subunit is 50s and sedimentation coefficient for small subunit is 30s. Whereas in case of large subunit of eukaryote, the sedimentation coefficient is 60s and the sedimentation coefficient for small subunit of eukaryote, it is 40s. Okay, and in prokaryote the large subunit have two rRNA, that is 23s rRNA and 5s rRNA, whereas in eukaryote there is three type of RNA, that is 28s rRNA, 5s rRNA and 5.8s rRNA. And in case of prokaryote, the small subunit have only one rRNA, that is 16s rRNA, whereas in case of eukaryote, the small subunit have 18s rRNA. So in case of prokaryote, remember the large subunit have 31 proteins, whereas in case of eukaryote, the large subunit is 49 protein. Okay, whereas in small subunit of prokaryote, there is presence of 21 protein, and in case of small subunit it is 33. So this is the comparison of the component of prokaryotes and eukaryote. Now let me told you a interesting thing, that is, inside eukaryotic cell we all know that mitochondria present, and if it is a plant eukaryotic cell then there will be a chloroplast, right? Now surprisingly, in plant chloroplast and mitochondria for both animal and plant, inside them ribosome is present. Okay, and the most surprising thing is, in the mitochondrial ribosome and the chloroplast ribosome, the sedimentation rate is 70s, which is similar to prokaryote, despite they are present in eukaryote. So, this information or this evidence simply suggested the idea of endosymbiotic theory. That is, mitochondria and chloroplast are nothing but microorganisms who have fused with eukaryotic organisms in earlier times, and then they evolved as mitochondria and chloroplast. And among them, mitochondria helping us to respire and chloroplast helping plants to photosynthesis, right? So that is the idea. So another important thing is, after detailed analysis of mitochondrial ribosome and chloroplast ribosome, it was observed that chloroplast ribosome have similarities with bacterial ribosome, and mitochondrial ribosome do not have that much similarities with bacterial ribosome as chloroplast ribosome has. Okay, so that is what here I have written. And remember, the ribosome present in mitochondria is known as mitoribosome, and the ribosome present in chloroplast is plastoribosome. And there are various other information present, but which is, I think, not very much important. You may read this if you want. So now another important part of ribosome, that is, ribosome may be classified as free or membrane bound. Okay, and that is the reason why we cannot consider ribosome as organelle, because remember, a organelle is the term which will be used for subcellular component having phospholipid membrane. But ribosome are classified on the basis of the presence of or absence of membrane, right? That simply suggests that all ribosome do not have any kind of, all ribosome do not have membranes, right? Some of them have and some of them do not, right? So that is why we are calling ribosome broadly as non-membranous organelle. That is very, very important. You may ask that ribosome, why we are not calling ribosome as organelle. This is very, very important question. So now, whether or not membrane will be present is totally depending upon presence of ER targeting signaling sequence in the protein which is synthesized in the ribosome. If ribosome synthesizing a protein having, so whether a ribosome have membrane or not is totally depending upon the ER targeting signal sequence present in that particular protein that is synthesized by ribosome. If ribosome are synthesizing the protein who are having ER targeting signal sequence, its spatial arrangement will be different from the ribosome who are having or who are synthesizing proteins who lacks ER targeting signal sequence. So that's the idea. So now remember, free ribosome can move in any part of the cell, only excluded the cell nucleus and other organelles, and they can synthesize proteins who are actually used by the plasma membrane or who are actually used by the cytosol. Okay, and another important thing, that is, free ribosome will not going to synthesize any kind of protein having disulfide bond, and the disulfide bond who are synthesized by oxidized cysteine residue. Why? Because remember, in the cytosol there will be high concentration of glutathione, and this glutathione is biologically active tripeptide and it is responsible for reducing environment. Okay, now try to think about this. If a protein which is having disulfide bond which is synthesized from oxidized cysteine residue, then in presence of reducing environment those oxidized cysteine residue will be reduced again. Okay, that causes the disruption of that protein. That is why free ribosome will not going to synthesize any kind of protein who have disulfide bond synthesized by oxidation of the cysteine residue. Another important thing, ribosome who are synthesizing proteins for some organelles will be membrane bound, and this type of ribosome will going to synthesize protein especially in a specialized region of endoplasmic reticulum, that is rough ear or rough endoplasmic reticulum. So after the synthesis of protein in the rough endoplasmic reticulum, the ribosome will going to insert that protein in the endoplasmic reticulum via vectorial synthesis, and then from the ear, it will be transferred to their preferred destination via secretory pathway. We have already discussed about the secretory pathway during our transport of protein, where I have told you that from the ear lumen, by the formation of vesicles, it will be transferred to cis Golgi, and from that it will be transferred to trans Golgi and then to lysosome or plasma membrane. That's how the pathway is going on. So that's the thing. So here we have learned that ribosome can be classified as membrane bound or free. Free ribosome will going to synthesize proteins which are actually play their role in the cytoplasm, right? And remember, free ribosome will not going to synthesize any type of protein who have disulfide bonds, and especially the disulfide bonds which are synthesized by oxidized cysteine residue. Why? Because cytoplasm have glutathione, and this glutathione is responsible for the reducing environment of the cytosol. And if any type of protein will be released in the cytosol having disulfide bond formed by oxidized cysteine residue in presence of reducing environment, it will be converted back into its reduced form, causing the disruption of that particular protein. That is why those kinds of protein will not be synthesized by free ribosome. Free ribosome will only going to synthesize proteins which are used up by the cytosol. Whereas membrane bound ribosome will going to synthesize those type of protein as well as protein who are used up by some type of organelles. And this type of protein will be synthesized in a specified region called rough endoplasmic reticulum. Ribosome, after synthesis of the protein, will going to insert that protein in the ear, and from that ear it will be transferred via secretory pathway. Okay, so that's the idea. Along with that secretory pathway, it may also use exocytosis for transfer of protein. So that's the idea. So I hope you can understand about the free or membrane bound ribosome, and it is totally depending upon which type of protein are synthesized inside. If that protein have ear targeting signal sequence, then that ribosome will be different from the protein who lack ear targeting signal sequence. And remember one thing, this free and membrane bound ribosome have difference in their spatial distribution. Okay, otherwise they are identical in their structure. So that's the idea. And here I have also told you about why we are calling ribosome as non-membranous organelle. We are not calling them as organelle because ribosome lacks phospholipid membrane. Not all, but some ribosomes, right? And that is why we cannot consider them as organelle, because organelles are subcellular component having phospholipid membrane. So from that concept, we can say mitochondria as organelle, right? Because we have discussed about the outer membrane and inner membrane, right? So that's the thing. So now let's talk about the synthesis of ribosome. So synthesis of ribosome in case of bacteria occurs in the cytoplasm through the transcription of the ribosome RNA genes operons. Okay, because there will be presence of ribosomal gene operon in the cytoplasm of the bacteria that will be transcribed, and synthesis of ribosome will occur. And in case of eukaryote, the ribosomal synthesis will be take place in against cytoplasm as well as a specified region of the nucleus, that is nucleolus. We will going to talk about nucleolus in details in our nucleus video. So what will happen? Ribosomal, if I going to use this picture, then it will be easy for you for understanding. You can see here we are synthesizing eukaryotic ribosome. That is why this is going on in nucleolus. There is a operon for ribosomal genes present. So what will happen? First, all the ribosomal proteins as well as the large subunit, small subunit, all things will be synthesized along with the rRNAs. Okay, then there will be assembly of those things, and near about 200 proteins will going to play together to assemble all those things to create the ribosome. Okay, so that is the synthesis. Now, at last, another important idea, that is, according to the RNA world hypothesis, ribosome is the first who have originated. But surprisingly, in the earlier times, it was constructed solely of rRNA. That means no protein was present earlier. And this rRNA have self-replicating capability. That is why in earlier times it was considered that ribosome was the thing who can self-replicate. Okay, and these rRNAs have all the informations which is responsible for structural as well as catalytic purposes. Okay, so that is the idea. So that is the video for our ribosome. So here what we have to remember, we have to remember this slide very carefully. That is the difference between prokaryotic ribosome and eukaryotic ribosome. An important MCQ, that in eukaryote, which organelle have ribosome who having similarities with prokaryotic ribosome, that is the ribosome in case of mitochondria and chloroplast. Then here the classification of ribosome depending upon whether membrane is present or not, and it is totally depending upon the presence of ER targeting signal sequence in protein which are synthesizing inside the ribosome. So that's the idea of our ribosome.